Okay, thanks for the previous two speakers, gave very detailed information. That's a lot of information to learn and also laid a solid foundation for my talk. And here I will switch gear to give you an overview of the OPQR testing and research to support GSP development. First, here's a message from Office of Pharmaceutical Quality, OPQ. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. So through this, here's the learning objectives of this talk. So through my presentation will inform you on FDA's own laboratory support on inhalation drug assessment. And you will understand the research efforts conducted at the FDA lab to support guidance development. And at the end of this presentation, I hope you can list several newly recommended in vitro studies as options to establish BE for inhalation products. First, okay, this topic is OPQR Labs support for GSP. So who are the OPQR Labs? So OPQR stands for Office of Pharmaceutical Quality Research. It's a sub-office under OPQ, and we are a lab-based office having 6 divisions. The inhalation lab and team is under Division 2, so we are located in St. Louis, Missouri. In our inhalation lab, we have a full line of equipment to do testing and research for inhalation drug products, including delivery dose uniformity testing for MDR and DPIs, Anderson cascading impactor, ACI, next generation impactor, NGI, for aerodynamic particle size distribution, APSD testing. And we have non-impaction fast imaging method instrument Supervisory to measure spray pattern and plume geometry. And we have laser diffractions, and we have various type of microscopy, including Raman microscopy, 3D X-ray microscopy, SEM, AFM, and also some other FTIR microscopy. And we have dissolution capacities and to perform dissolution, diffusion, and permeation. So what do we do at OPQR Labs? So at OPQR Lab, first we are support regulatory actions. So we provide product testing and a method validation program to support NDA and ANDA reviews, and also support other type of regulatory actions. And we also conduct proactive research, first to evaluate emerging technologies for characterizing inhalation product performance. And we're also involved in develop novel methods to establish better in vivo in vitro correlations. And in the last few years, we put a lot of effort to conduct research to support a product specific guidance, PSGs, and especially focus on developing method for alternative BE approaches, including the 5 areas: realistic APSD, laser diffraction, particle morphology, and we're also exploring evaporation rate and the velocity profile for MDIs, and we'll also develop clinical relevant dissolution method for MDI and DPIs. So in this talk, I will give you some examples on the first 3 areas. First is on realistic APSD, which the previous 2 speakers already talked a lot. So I will focus on more details for what you see in the lab. So traditionally, APSD was measured by cascade impactor. And the cascading impactor collect respirable aerosols with particle size less than 9 microns. So ACI is a stack of metal jets and plates to collect those particles. And between the cascading impactor and the drug product, there's a sample induction port, which shows in this picture is a metal piece with 90-degree straight bend. So it's nothing like the human anatomy. So the first area to improve to develop realistic muscle models, as the previous speaker mentioned, is to using bio-relevant muscle models, such as a commercial available Alberta idealized throat, short as AIT, or models developed by Virginia Commonwealth University, VCU. And the VCU models, the 3D profile is available online. You can print it out using 3D printers using different materials. So the second area for the realistic model, APSD testing is to utilize realistic breath simulation. And here the scheme is what we do in our lab to set the equipment up. Traditional way is just using a pump directly connected with a cascading impactor. So that only provides you a constant airflow. So it's not simulate the human inhalation profile. And by adding a mixing inlet and also utilize a breath simulation equipment and incorporate compressed air, we can set up simulated flow through the APSD testing. So by combining the bio-relevant muscle models and the realistic breath profile, a better RV-RVC are expected. Here is an example for our previous study. And this study was published in 2019 on AAPS Pharmaceutical. So the study is to investigate solution MDI and the suspension MDI using different type of throat models. So our result shows, first of all, the comparison between in vitro and in vivo result are product specific. And for the solution-based MDI, all the muscle models shows close match to the clinical data. Even the USP throat shows close to the clinical result. However, for the suspension MDI, the results are mixed. Overall, the bio-relevant muscle models here, VCU models, have a better chance to give you a better RV-RVC. So second area we spend quite a time on is to investigate the feasibility of a laser diffraction to be used as an alternative or orthogonal APSD method. Laser diffraction is a mature technique in a lot of industries. So when a particle passing through a laser beam, a larger particle will give a small diffraction angle, while a small particle will give you a larger diffraction angle. And the pros of this technique is it provides a rapid measurement. Normally, it delivers the result instantly. However, there's cons. First, laser diffraction measures volume-based PSD. Although it can be correlated to APSD, but still it's not a direct measurement. Second, is more important, laser diffraction is not able to differentiate API and excipient particles. So therefore, for complex drug formulation with multi-component, laser diffraction cannot be used as an alternative method for APSD. However, for some simple formulations such as solution or dry powders with only API presented, laser diffraction have a very good chance to be an alternative APSD method. So there's a 3 new product has been studied using laser diffraction. First is the Spriva Respimate. It is a soft mist inhaler. And the soft mist inhaler is a kind of product between MDI and nebulizer. The device is very similar to an MDI device. However, the formulation in the aqueous solution is similar to nebulizer. Since the formulation is a homogeneous solution, so their droplet size can be studied as a potential alternative for APSD. And our results, laser diffraction results shows a clean measurement of the aerosol with a medium particle size around 3 to 5 microns from different distance, which demonstrate the laser diffraction could be have a very good chance to be an alternative method for APSD for this drug product. The second example is a breath-actuated nasal powder on Vitro axial. For this product, it just have a special design, and the airflow is coming from the patient mouth to blow in the air into the device. And the airflow bring the powder out, introduce the powder into one nostril of the patient, and the airflow coming out from another. So the drug formulation here is around 15 milligram of API powder only. Since the aerosol are API particles only, so it also have a good candidate for laser diffraction to be an alternative APSD method. And our result demonstrated this point, and with a good clean measurement of laser diffraction, shows the medium particle size is around 30 micron. And the laser diffraction not always give you perfect result. And for the third example, is a breath-actuated thermally generated inhalation powder. So for this product, we had some trouble. The main point is most of the drug product was released at the first half seconds of the aerosol procedure, and it's highly condensed. The concentration is so high, and the laser diffraction measurement was not valid at the concentration. So the detail of this study has been reported in the last year's SBIA workshop, and more details were published at the RDD 2024 proceeding. And the conclusion is the laser diffraction was not recommended as an alternative method, APSD method for this drug product, due to the limitation of the high concentration of API particles. So the third area I want to talk about is to using MDRS, morphologically-directed Raman microscopy, to characterize particle morphology and API-specific particle size distribution. So to demonstrate Q3 sameness, the particle morphology and the API particle size distribution is critical. Here, when we're talking about particle morphology and the particle size distribution, we're talking about API, not excipient. However, the challenge are apparently for most of the product, API and excipient particles coexist in the formulation. And sometimes more than one API will be presented, and the API may have more than one polymorphic forms. The traditional techniques, such as laser diffraction we just mentioned, they cannot differentiate chemical identities of the particles. So Raman microscope is a good candidate for characterized particles, API-specified particle size and the particle morphology. However, normal Raman microscope is a very time-consuming process. And the MDRS provide a special strategy, specially designed for particle sizing characterization by implementing a morphology screening procedure before Raman conformation. So the basic idea is that morphology filters will exclude as many as excipient particles as possible before time-consuming Raman measurement. Therefore reduce the measurement time by 10 folders. At our lab, our research is focused on development, optimization, and the validate of MDRS method on different type of drug formulations, such as nasal spray suspensions, dry powder inhalers, transdermal systems, ophthalmic ointment, and the cream. And we have 2 publications in 2019 and 2021 on nasal spray suspensions. And we also have a poster on MDRS application on creams in this SBIA. Hope you have time to visit our poster session. In summary, inhalation drug testing and research is an integral part of OPQR's work. And our OPQR lab-based research programs provide data to support guidance development, facilitate evaluation of generic drug applications, and allow for risk-based assessment of new drugs. Here, I want to acknowledge our past and current inhalation team members and our collaborators from OPQ, OPQA, and OGD, ORS, and OGD, OB. Finally, challenge questions. First, which city is the OPQR inhalation lab located? A, Rockville, Maryland. B, Silver Spring, Maryland. C, Washington, D.C. D, St. Louis, Missouri. The answer is D, St. Louis, Missouri. Okay, that's an easy one. Second one is not difficult either. So, which of the following technique method is not considered as an alternative B approach for assessment of inhalation drugs? A, realistic APSD. B, solid state NMR. C, clinical relevant dissolution. D, particle morphology. The answer is B, solid state NMR is not an alternative B approach for inhalation. Okay, with that, I'm wrap up the presentation session. I will welcome the 2 speakers and the 2 other members to come here to host a Q&A panel session. Thank you.